Hello, everyone. My name is Houda Chamseddine, and I am a student here at the University of Texas at Arlington in the Department of Psychology. And today I will be presenting sort of an extension of a previous submission, and it'll be exploring sex differences and acute condition morphine withdrawal. Here's just the overview of my poster. And just for a bit of background, an estimated 26.8 million people live with opioid use disorder globally, and that estimate is highest here in the United States. Over 81,000 deaths occurred as a result of drug overdose in the United States between May 2019 and May 2020, and that's the highest recorded number in just a 12-month period, according to the CDC. Now, the primary cause of drug overdose deaths during this 12-month period looks to be opioid use. Now, what we know about opioid use is that it is true that a greater number of men report addiction to opioids compared to women. However, women progress from this casual use to addiction at an accelerated rate when compared to men. This is something called telescoping. There are also behavioral studies that have exposed some of the sex differences present in opioid addiction, reward, and dependence. However, some of these studies are really lacking, especially when it comes to withdrawal, because much of what we know about opioid use disorder, and more specifically withdrawal, is based on data from exclusively men and male samples. To date, there's been little investigation into the influence of sex on these environmental cues, specifically when paired with drug withdrawal. So the experiment aims to use conditioned place aversion as a method of exploring the influence of sex on naloxone-induced place conditioning. So the overarching purpose of this experiment was to, of course, explore whether sex or estrous cycle differences exist during acute opioid administration and naloxone precipitated withdrawal by using the CPA paradigm. There were three hypotheses that we had. First, intact females would be more sensitive to the condition aversive effects of naloxone than intact males. Second, among intact females, aversion intensity would vary across the estrous cycle. And of course, third, overectomized females treated with estradiol would express higher levels of aversion compared to overectomized females treated with peanut oil. And here's just a figure demonstrating the CPA paradigm and sort of a timeline. And I did want to go over what conditioned place aversion is. It's a classical conditioning paradigm used to test negative motivational effects of objects or experiences. So in this case, that negative motivational effect would be drug withdrawal. And the apparatus consists of 3 chambers, 2 that are designed for the animals to discriminate between them based on color and the floor texture. So you can see here a black chamber and a white chamber. One floor has bars, and the other is more of a grilled texture on the floor. Then there's a middle chamber to connect the two, which is the neutral chamber. So this place conditioning procedure involves 3 phases: a pre-test phase, the conditioning phase, and a post-test phase. If the withdrawal effect is motivationally significant, the animal, we expect them to spend less time in the chamber associated with that aversive effect. So the idea here is when the unconditioned stimulus is aversive, the rat will avoid the compartment that contains cues associated with it. So, what did we find? In this figure, we see red bars represent time in the naloxone paired compartment, and blue bars represent time spent in the saline paired compartment. Male data are checkered, and female data are striped. And of course, data are expressed as CPA scores, which was calculated from the difference of time spent in seconds in each compartment during the pre-test and post-test. So we took the post-test with the pre-test and subtracted it from the amount of time spent during the post-test. Now, it's true that both male and female animals achieved aversion, but there was no significant difference between the aversion scores of males and females. So next we looked at just females across estrous cycle. So looking at CPA scores across the ester cycle, we again see there was no significant difference between females with high and low levels of estradiol during the naloxone conditioning phase. But females with high levels of estradiol during the naloxone conditioning phase displayed significant CPA, while those with low levels of estradiol did not. Now, it's important to take in mind that our sample size is very small here, so we wanted to sort of extend these findings and look at overactimized rats where we could control for levels of estradiol using exogenous administration. So here again, we have our intact females displayed as CPA scores post-test minus pre-test across 2 days of conditioning, which is here on the left, and then 4 days of conditioning, which is shown on the right. All females spent significantly less time in the naloxone paired chamber, whether they were intact or overactimized, than the saline paired chamber. There were no significant differences found between intact, overactimized, and overactimized animals that were treated with hormone treatment. So peanut oil or hormone treatment showed no significant differences. So, based solely on the behavioral data collected, it does appear that male and female rats display no differences in their sensitivity to the conditioned aversive effects of naloxone-induced withdrawal. Females with high estradiol did display significant CPA, while those with low levels of estradiol did not for intact females. However, when we followed that up with the overactimized females and treated them with exogenous estradiol, did not appear to have any impact on aversion scores. So, where does that leave us? Overall, I do believe these findings still do underscore the importance of assessing endogenous ovarian hormones and their effects on mediating response to drugs of abuse. It's also important to consider convergent sex differences, where males and females might exhibit the same behavioral response, but the mechanism underlying that response differs. So this is where, in terms of future direction, our immunohistochemical analysis will be important. We're also looking to characterize not only the motivational and affected related measures of withdrawal, but we're looking at sex differences as somatic components of withdrawal during chronic opioid use. Thank you.